So quickly, a little bit about myself. I primarily work on modeling human visual cortex, especially the higher visual cortex. So human vision divided into early and higher. I focus on the semantic and categorical perception regions in the brain. So my field primarily, if I had to characterize it, is AI for neuroscience. Currently, I'm an assistant professor in data science and psychology at HKU. Previously, I did my PhD in machine learning and neurocomputation at CMU, and before that, computer science at MIT. So today, quickly, I'm going to talk about how humans perceive and understand the world through vision, through light, and how we can understand higher visual semantics in the brain. Then I will talk about computational approaches to understanding higher vision in the context of decoders and encoders in vision science, the limitations of current encoders, and how we can design encoders as conditional function inference. And finally, I will quickly discuss some insights and future directions. So why should we study human vision? I usually give this talk to computer science people, and they were like, okay, we have these great neural networks. Machines can see better than the brain. Why should we study human vision at all? I can tell you, these people are absolutely wrong. Humans and computers, they use similar features for scene processing. And the hierarchical features from early to higher visual cortex is similar to the feature integration that you see in these neural networks, convolutional neural networks, vision transformers. And the evolutionary constraints of, for example, energy use, few-shot learning, these are insightful for machine learning as well. And finally, I always tell my students, it's easier to copy and take insight from evolution than invent from scratch. So how does the human visual system work? Well, first you have retinas. So retinas are located in your eyes. That's the first stage of visual processing. The retina connects to early visual cortex, which acts as a simple feature extractor. And finally, that builds up a connection to higher visual cortex. And that is responsible for semantic and categorical perception. Since this is only 20 minutes, I'm going to go over this really quickly. The human eye is definitely not a camera. There's a constant system of feedback, self-correction to adjust focus, to adjust what you look at. But your brain is actually doing a ton of processing. It makes you think that you're seeing a consistent view of the world, even though you're constantly blinking, you have a blind spot, you can't really see in full color, but you don't know that. Your brain kind of tricks you. And the first stage of visual processing is early visual cortex. And the early visual cortex is really responsible for simple features. So for example, edges, orientation, gradients, that kind of stuff. So very simple features that a student, for example, could design. And the brain really progressively builds up a set of features from V1, so that's primary visual cortex, to V4. So that's some people consider that the last stage of early visual cortex. And this was the results that really won the Nobel Prize in 1981 in medicine. I can tell you what's going to be seen here. It's just a single white ball moving across the screen. And they showed this to a cat, and they won the Nobel Prize. And that's my dream as well. But this was, 50 years ago. So the brain builds up a hierarchy of representations from early visual cortex to higher visual cortex. And finally, in higher visual cortex, this is the back of your brain. And this is generally believed to be semantically selective. So you have brain regions that is activated when you see bodies, when you see faces, places, words, food, or objects, or even multiple different categories. And what I mean by selective is a region that is selective to food is activated when a person sees food. And this is another very famous neuroscientist in the 1960s, Charlie Gross. And he spent a lot of time, he didn't get any results. And finally, he went to the store and he bought a toilet brush, showed it to the monkey, and the monkey neuron activated. And he became a very famous neuroscientist. That's my dream as well. But we can't all go and buy toilet brushes, right? So how can we understand visual selectivity via data-driven techniques? So given some region of the brain, how do we know what activates it? The world is huge. There's so many different objects. We don't really know a priori what activates a given brain region. So that's why my work is on computational understanding of visual cortex. And my goal is to understand object recognition and category recognition as how it is facilitated by higher visual cortex by leveraging these data-driven computational tools without using handcrafted stimuli. So before I go any further, I really want to talk about encoders and decoders. So this is like the bread and butter for computational neuroscience. The encoders map from stimulus to brain activity. So you see an image, the encoding model predicts what your brain activation is like, while decoders do the reverse. So it maps from brain activity to the image or some text or something like that. But encoders are really useful as proxy models of the brain, while decoders are useful in telling you what information the brain is currently processing. So I'm not going to talk about all of this work. This was adapted from some of my other slides. I will talk about the first work, brain diffusion for visual exploration, brain dive, and then I will transition to talking about meta-learning and in-context transformer model of human higher visual cortex. And traditionally, people really looked at selectivity by running these experiments. They show a human or a monkey or some other animal some images within a category, and then they show these images not in that category. And essentially, they look at the neural activity and they do a subtraction. And they say, okay, face activity is higher here, house activity is higher here, so this is the face region, this is the house region. But of course, people have also tried to use computational approaches to look at human and animal perception. For example, the work from UT Austin, Tolias's work, MIT's DiCarlo's work, Carnegie Mellon's Leila Wahebe's work. And essentially what these works do is they train a model of human visual cortex, and then they do gradient-based optimization in image space. And using these methods, they can find really these images that activate different brain regions. However, this approach does not work well for higher visual regions. This is from Harvard's Margaret Livingstone, and she worked on monkeys, and she showed that by constraining the output using large visual models, they can generate images that she claims are very similar to monkey faces. And really, people have been building up using more and more complex models, and they find these images that activate different brain regions, like the fusiform face area, extrastriate body area, place area, et cetera, et cetera. And our work proposed this brain dive approach, which I'm not going to bore you with the math, because there was a lot of math. And essentially what it does is it can target different brain regions and generate images that can activate these brain regions without you needing to know ahead of time what activates a given brain region. So I can say, okay, activate this face region. I'm not going to tell you it's a face region, and it will generate faces. And we actually did some MRI scanning, and we compared these generated images against natural images, and we find that these generated synthetic images are actually more highly activating. So this potentially has some use in terms of therapeutics. If we want to target some brain location and we want to activate it through visual stimulus, this is one potential approach. And we looked at this across face regions, body regions, word regions, and food regions. And with the exception of food regions, our approach consistently achieves higher activation. But of course, there is a downside. So all of our prior approaches relies on training one model per subject. So subject 1, I have one model, one encoder. Subject 2, I have one encoder. But how is it possible that we can generalize and we can learn from subject 1 to 2 to 3, and for example, inform our decision on how we want to design the encoder for subject 4? However, this is actually quite tricky because every human is different. Some people are missing half the brain, although you can't tell. Some people are left-handed, right-handed. Young children are very different from adults. How far you are in the developmental progress. For example, if you're a young child, maybe you don't have the word region developed yet, but when you're a teenager, you do have a word region. So we really thought about this, and we thought, how can we design these encoder models that can generalize across subjects? So just by looking at subject 1, 2, and 3, for example, at different ages or different left or right-handedness, can we inform the design of encoder for subject 4? And our goal is to recover the true response function from few-shot observations. So we framed this as a hypernetwork learning problem. So our question is, how can we use information from 1, 2, and 3 and use it to inform, adapt to new subjects? And it doesn't have to be just a new subject. It could be a new MRI scanner. It could be a new protocol. It could be a new voxel size, different stimuli. And we want to achieve all of this without any fine-tuning. So we don't want to do any expensive fine-tuning. So our approach is to leverage in-context learning from these large language models. So large language models are trained on next token prediction, NTP, but they really can solve very diverse tasks via in-context conditioning. So in-context conditioning, you can understand it as a type of meta-learning. So instead of telling the network, this is input A, we want output B, we say this is input A, input B, output B. Now I have input C, what is output C? So we're teaching the network learning to learn. And this is a type of demonstration-based learning. And effectively, what we're doing is encoding as function estimation. So we're assuming that we have a set of different functions. Each function represents a mapping function from visual stimulus to a given brain region. And our goal is to invoke this function and achieve few-shot in-context adaptation to novel subjects. And our optimization process is learned using meta-training and in-context learning, a combination of both. And we proposed this model, we trained a ton of models, and we find that our model can achieve far better performance than simple linear regression or even anatomical baselines. And we demonstrate consistent performance scaling as the context size increases, and even zero training adaptation to novel scanners with different protocols. So we can also achieve segmentation of visual cortex with just 100 images. So with just by showing you 100 images, I can characterize how your visual cortex is organized, which is, I think, is actually quite an impressive achievement. My question is, how can AI help crack the code of brain function? And in this work, we proposed a foundation model of neural activity by meta-learning an in-context transformer model using 100 million parameters, so a very small model. And this is vastly more data efficient when adapting to a new subject, and we see consistent performance improvements as the test time context increases. And we find it actually performs best when it is conditioned on diverse stimuli. So you don't want to show it 100 images of faces. It performs best if you show it very diverse and different stimuli. And to summarize, we have a set of tools to help practitioners explain and understand how brain functions in different subjects. And our latest work leverages these in-context transformers to model the brain. So a quick acknowledgement to my collaborators and students, and thank you so much. You can check out my work at our website, and I will take any questions.